So now moving on to proteins, just like we did for carbs, you can look at them in terms of sizes. So you've got proteins broken down into oligopeptides, then digested into amino acids for absorption, right? However, we can also, in some cases, absorb them intact. And so one of the examples of this is neonates, so infants. They drink colostrum, which is breast milk, and they absorb intact protein through endocytosis. Also, we can absorb intact proteins for immune response by endocytosing antigens and presenting them to immune competent cells. So let's look at that here. So essentially, proteins are digested and absorbed into amino acids, but infants can endocytose intact proteins from breast milk, and some proteins are absorbed intact for an immune response. What are the sources of proteins? Well, sources of protein, 50% diet, so exogenous, 50% endogenous, so that's from inside us, and the endogenous proteins, they're from cells, desquamated cells, hormones, and enzymes. Now, one thing that you have to understand, you have to get your head around this to understand the rest of it, essentially, is that there's 4 routes of protein digestion and absorption. Okay? So proteins, when you eat them, they're digested by luminal proteases. So these are proteases inside the gut lumen, and they're digested to oligopeptides. And once this happens, they can one of 3 things can happen essentially, right? They can either be further digested by the same luminal proteases into amino acids. They can be digested by brush border enzymes rather than the luminal proteases, instead digested by the brush border enzymes on the brush border of the enterocytes. Or through the PEP-T1 hydrogen cotransport channel, or cotransporter, the oligopeptides can be absorbed into the enterocyte. So let's look at the first route. So luminal proteases digest the proteins to oligopeptides and further digest them to amino acids. The amino acids that are imported across the apical membrane, and this is there's a few different ways in which they can be absorbed across the apical membrane. And then what happens is once they enter the enterocyte across the apical membrane, they're then effluxed out and into the blood, across the basolateral side into the blood. So that's the first route. The next route is that the luminal proteases digest the oligopeptides, digest the proteins to oligopeptides. The oligopeptides are then further digested by brush border enzymes, and the resultant amino acids are imported across the apical membrane, effluxed across the basolateral membrane and into the blood. So that's the second route. The third route is that the luminal proteases digest the proteins into oligopeptides. The oligopeptides are taken up into the enterocyte rather than being digested. They're taken up through the PEP-T1 hydrogen cotransporter into the enterocyte. They're then inside the enterocyte. They're then digested by cytosolic, which means enzymes inside the cytoplasm of the enterocyte. So they're digested by cytosolic enzymes to provide amino acids which are already in the cytosol of the enterocyte, from where they're effluxed across the basolateral side into the blood. But what can also happen once the oligopeptides have been transported into the enterocyte via PEP-T1 cotransport is that from there the oligopeptides intact are absorbed into the blood. So now looking at the digestion of the peptides. So the luminal digestion, the first thing that happens is that in the stomach, gastric chief cells, which are the big cells of the stomach, remember, they secrete something called pepsinogen. The ogen implies it's a proenzyme, so pepsinogen enzyme. And because of the low pH, it's activated to pepsin, which is an endopeptidase, which I'll explain later. And what that does is it partially digests 10 to 15% of the proteins. Now, what stimulates the gastric chief cells to secrete pepsinogen? Well, it's acetylcholine, so vagal stimulation from whatever that might be, gastric distension, and that causes acetylcholine to stimulate chief cells to release pepsinogen, activate it to pepsin, and partially digest 10 to 15% of the proteins. One more thing is that the pepsin also activates pepsinogens to form more pepsin. So gastric cells produce pepsinogen for protein digestion. The next thing that happens is, so continuing on from luminal protein digestion, the pancreatic acinar cells, they produce zymogens, which you'll recall from the lecture on the pancreas. And zymogens are activated to either exo or endo peptidases, so peptidases, protein enzymes, right? So these pancreatic acinar cells, they secrete 5 proenzymes, which are zymogens, 5 zymogens, including trypsinogen. And when trypsinogen enters the duodenum, enters the small intestine, it's activated by small bowel enterokinase, which is a brush border enzyme. And what enterokinase does is it activates the trypsinogen to trypsin, and trypsin activates the other enzymes, activates the other proenzymes to form enzymes. So let's just take a moment to think about what stimulated the pancreatic acinar cells to secrete the zymogens. Well, remember the main stimulant of pancreatic acinar cells is cholecystokinin. And where does the cholecystokinin come from? Well, it's essentially to do with fat. So if you have fat, then the luminal cholecystokinin releasing factor is not digested by the digestive enzymes. And therefore, luminal cholecystokinin releasing factor stimulates the duodenal eye cells to release cholecystokinin, and cholecystokinin stimulates the pancreatic acinar cells to secrete zymogens. Another stimulant of the pancreatic acinar cells is acetylcholine, and acetylcholine and cholecystokinin act through the PKC pathway, whereas secretin and VIP, they also stimulate acinar cells through the PKA pathway, adenyl cyclase PKA pathway. Where can VIP, vasoactive intestinal peptide, come from? Well, it can come from a pancreatic tumour, such as Werner-Morrison syndrome. Where does secretin come from? It comes from duodenal S cells in response to acid. So once trypsin has activated the other enzymes, the other proenzymes secreted by the pancreas, you can get 2 peptidases essentially. There's endopeptidases, and what they do is, you can guess by the name endo, they cleave the protein from within. And this produces oligopeptides, which are larger. And this is most of them, 70% of them, oligopeptides. However, the pancreatic peptidases, they can either be endo or exopeptidases, and the exopeptidases are carbo, A, and B. And what the exopeptidases do is they cleave the protein ends. So if you cleave the ends, they produce amino acids, which is 30% of the product of the luminal protein digestion. And if you recall back to the sort of the diagram of the different routes of protein, these oligopeptides that are produced, there's a few different things that can happen to them. They can either be further digested by luminal digestion to amino acids. They can be transported into enterocytes via PEP-T1, co-transporter with hydrogen, or they can be digested by brush border peptidases.